Hello everyone, welcome to Ritika's online teaching academy. In the previous lecture, I have talked about the initiation of replication in eukaryotes. Today, I will talk about the elongation process in DNA replication of eukaryotes. Okay, now I am going to share my screen with you. So it is lecture 7 in molecular basis of inheritance, and it is replication in eukaryotes about the elongation process. I am Dr. Ritika Gupta, and today I will discuss about the elongation process. So first we will recall the various enzymes and proteins involved in DNA replication in eukaryotes and compare with the E. coli proteins and enzymes. So DNA protein which is found in E. coli, its comparable protein in eukaryotes is ORC, which is called as the origin recognition complex. And I have discussed about it in more detail in the previous lecture. So it recognizes the origin of replication. The second one is the DNA B protein. DNA B protein, its analog in eukaryotes is MCM, or we can say mini chromosome maintenance protein. It acts as a DNA helicase. So in eukaryotes, DNA strands are separated by MCM proteins and unwinds the double-stranded DNA. The DNA-C protein, which is unknown or uncertain in case of eukaryotic proteins, its function is to load helicase onto the DNA. Next protein, which is called as primase in case of E. coli, it is also called as primase in case of eukaryotic proteins and enzymes, and it synthesizes the RNA primers. So RNA primers are synthesized with the help of the primase enzyme in eukaryotes. Gyrase in E. coli is termed as topoisomerase 1 or 2 in case of eukaryotic proteins. And what is their function? They relieve the positive supercoils ahead of the replication fork. So we know that the DNA strands unwinds. So when it unwinds, it will create a tension in the DNA strand. So that tension will lead to the generation of supercoils, and those supercoils can be relieved by the topoisomerase enzyme in case of eukaryotes. If it is not relieved, the DNA replication would not occur. Then single-stranded DNA binding protein in case of E. coli are called as replication protein A or RPA in case of eukaryotes. Its function is to maintain DNA in single-stranded state. So in order to replicate both the parental strands of DNA, the two strands must be separated with the help of helicase. After that, in order to prevent it from resealing, there is another enzyme or protein called as replication protein A, which maintains both the separated strands of DNA in the single-stranded state in order to carry out the replication process properly. Next is DNA polymerase third core. So DNA polymerase 3 in E. coli is termed as DNA polymerase delta or epsilon in case of eukaryotes. So in case of eukaryotes, it is DNA polymerase delta or epsilon, and delta and epsilon functions are similar. What is their function? They are the primary replicating enzymes, means they are the DNA synthesizing enzymes which synthesizes leading strand on one of the parental strand and Okazaki fragments on the other strand, and that strand is called as the lagging strand. And also along with the DNA synthesizing activity, this enzyme is also having a proofreading activity. If any incorrect base is inserted, that would be removed by the proofreading activity of this enzyme in case of eukaryotes. Now the beta subunit in case of E. coli is called as the Pcna, or also known as proliferating cell nuclear antigen. So in the case of E. coli DNA replication, we have studied about the beta subunit. So it is a ring-shaped subunit. In eukaryotes also it is a ring-shaped subunit of DNA polymerase holoenzyme. Holoenzyme means it is made up of many subunits, and this holoenzyme is in ring shape that clamps replicating polymerase to DNA. It means that the polymerase which is having DNA synthesizing activity, it needs with the help of a clamp, and that clamp is what? Pcna. Means Pcna help that replicating polymerase delta or epsilon in case of eukaryotes to hold on to the DNA. Now its function is similar to the polymerase third in E. coli and polymerase delta and epsilon in case of eukaryotes. So next subunit is gamma complex. Gamma complex is called as the RFC or replication factor C in case of eukaryotes, and its function is what? It is also a subunit of DNA polymerase holoenzyme that load the clamp onto the DNA. Okay. Next is unknown protein in case of E. coli, while there is a protein called as polymerase alpha in case of eukaryotes. It synthesizes short DNA oligonucleotides as a part of RNA DNA prime. Now polymerase 1 is called as the FEN1 in case of eukaryotic proteins, and it removes the RNA primers. Polymerase 1 of E. coli also fills the gap with DNA. Means here FEN1 act as only will remove the RNA primers. It will not have the gap filling activity. While in case of E. coli, polymerase 1 is do the dual function. It removes RNA primers as well as fills the gap with DNA. In E. coli, DNA ligase is also termed as a DNA ligase in case of eukaryotic proteins, and it seals the Okazaki fragments into the continuous strand. Next is what is the requirements for the replication system. So there is a common requirement of enzymes and proteins in the eukaryotes as well as in the prokaryotes. So here we are studying the eukaryotes. So here these are the list of the main enzymes or proteins involved in the case of eukaryotic DNA replication. The first one is helicases, second one is single-stranded DNA binding proteins, third is topoisomerases, fourth is primases, fifth is DNA polymerase, and sixth is DNA ligase. So these are the main proteins or enzymes as I have discussed in the previous table as well. Now, to understand in detail about the eukaryotic replication, it comes from the study of viral DNA, particularly SV40 DNA. So, in order to study the in vitro, the activity, or you can say the proteins of the eukaryotes involved in the DNA replication, we will study the viral DNA, particularly the SV40 DNA. So viral helicase is called as a large T antigen, which induces a strand separation at the SV40 origin of replication. So here SV40 DNA, we are taking it as a model for in order to study the eukaryotic proteins. So in SV40 there is a large T antigen which induces a strand separation at the origin of replication, and its function is to unwind the DNA as the replication fork progresses. And in the previous table, that protein which is involved in the unwinding of DNA is unknown in case of eukaryotes. Therefore, we are studying or this DNA helicase. We are comparing it with its activity with the large T antigen found in the SV40 genome. So DNA polymerase delta and epsilon, they are the primary DNA synthesizing enzymes. Now with the help of this figure, we can understand the replication process in eukaryotes in more detail. Here you can see the leading strand and the lagging strand, and these two strands are separated with the help of the large T antigen. Here you can see here this helicase where it is what large D antigen in SV40 genome. So it is separating or unwinding both the strands of the DNA. It is a ring-shaped structure. Now, the another protein or enzyme which is what RPA. What this RPA means, replication protein A. So what it does, it is acting as a single stranded DNA binding protein, means it maintains the both the strands in the single stranded state. So on both the strands you can see RPA is there. Now the next protein, which is what Pcna. Pcna is what uh, polymerase chain nuclear antigen. Proliferating cell nuclear antigen. What it does, it helps in loading or clamping of polymerase delta along with the RFC replication factor C onto the leading strand as well as onto the lagging strand. So it is helping both the polymerase delta as well as the RFC protein. Okay, so next is what, with the help of the main enzyme which is what polymerase delta, the leading strand synthesis as well as the lagging strand synthesis continues. But here you can see, or you can also recall from the previous lectures, that the leading strand synthesis is towards the movement of the replication fork, while the lagging strand synthesis is away from the movement of the replication fork. So here the another protein, or and also we know that on the lagging strand Okazaki fragments are synthesized, or we can say the DNA synthesis on the lagging strand is discontinuous or is not in a continuous manner. It is in a short pieces of DNA which are called as the Okazaki fragments. So in order to synthesize those short segments of DNA, primer is also required. So this polymerase alpha along with the primase enzyme will synthesize the primer, RNA primer. So this RNA DNA dimer would be synthesized, and at the 3' end of the RNA primer, polymerase alpha will add nucleotides to it. Okay, now here in the next picture we can see that lagging strand synthesis, in order to maintain the steady synthesis of DNA on both the strands as the fork moves, there is a formation of a loop on the lagging strand. So it is a necessity in order to make the DNA replication steady, there is a loop formation on the lagging strand. Okay, it will bring the polymerase delta along with Pcna and RFC factor to synthesize the DNA on the lagging strand as well. So in this way, the DNA is synthesized on both the lagging as well as the leading strand. And here also in the case of eukaryotes, the synthesis of DNA is semi-discontinuous, means it is continuous on the leading strand, while it is discontinuous in the case of lagging strand. So hence we are saying it is that the DNA replication is semi-discontinuous in case of eukaryotic replication. And here this brown color shape you can see, it is a topoisomerase. As the replication fork moves, that torsional strain or supercoils is removed by the with the help of the topoisomerase enzyme. Okay. So this now here we can see that DNA ligase and FEN1 are there. What is their function? We know already. FEN1 will act as an endonuclease, or we can say it will remove the primers which are now not required after the synthesis of the DNA on the lagging strand. So they are removed with the help of FEN1 endonuclease, and the gaps are sealed with the help of the enzyme called as the DNA ligase. So this is all about the elongation process in eukaryotes. In the next lecture, we will study about the termination or in DNA replication of eukaryotes. So in order to get those video lectures, please do subscribe and like my channel. Okay, thank you.